So in my previous video we were discussing about the asexual reproduction, and we have already discussed the binary fission, right? So now so the next topic we will study is basically Conidia. Now basically what is this? Conidia are basically they are formed, mostly they are formed in the fungi, but they are also formed in the bacteria too. Basically they are the spores like structure, or you can say they are basically spores. Basically they are formed on the filamentous bacteria. The bacteria must be filamentous, right? On the filamentous bacteria. For example, we have streptomyces. These are the example of the bacteria which have filament like structure. So they are only present on the filamentous bacteria. For example, actinomycetes, right? They are like, these are both an example of the bacteria in which the filament are present, and these spores are present on these. Basically it is present on the filament, right? So consider that this is a filament, and here we have the spores. We have these spores. Basically these are present at the transverse septum. These are present at the transverse septum of the filament, right? Okay, basically the filament which bears these conidia, these filament which actually bears the conidia is known as conidia force, right? So basically the filament which bears these conidia, these spore like structure, are known as conidiophores, right? Basically like after maturity, these conidia basically detach. They will detach and they will form a new bacteria. Right? Right. These spores will detach after the maturity, and this forms like this individual spore will form a new bacteria, right? Basically there are a very large number of conidia are formed by these filamentous bacteria. Basically there is bipedal succession. Now what does that mean? Basically bipedal succession means like at the apex, like at the above side, the older spores will be present, right? And the near the filament, the new spores will be present. That is known as bipedal succession. Like at the apex the old spores will be present, and the near the filament the new spores will be present. It is like the petal. Have you seen a petal formation? How the new petal is towards the you can say towards the flower, and the old one petal is away from the flower. This is like that. That is basically bipedal succession, right? Now we have another kind of asexual reproduction, which is like we have done with binary fission. Second was conidia, and third is basically budding. Basically the budding. What happens? Consider this is a bacteria. The bacteria, and it has its own genetic material, right? Everyone have its own genetic material. Basically what happens, there is an extension which is extended from the cell, from the bacteria. Basically it is a small extension, and after some time it matures. Basically it begins to grow. It grows in volume, grows in size, in volume and all this thing. Basically, this extension, or you can say this bulge like structure, is known as pretuberance. This structure is known as pretuberance, right? Now what will happen, this is the genetic material. This genetic material will get copied. And it will enter into this spore. Now this pretuberance is basically is bud. Now after the maturity, this pretuberance will deattach and it will form a new bacteria. And as you can see, there is an unequal distribution of the cytoplasm, right? There is unequal distribution. This pretuberance will form a bud, and this bud will deattach from this bacteria, right? Basically, this budding is seen in some planktomycetes, right? In some planktomycetes. This has been seen in some planktomycetes. For example, we have cyanobacteria. Like this budding has been seen in some planktomycetes, or you can say cyanobacteria. Like binary fission is very common, like it happens in every bacteria. Now this conidia happens in the filamentous bacteria, and this budding happens in basically in some cyanobacteria. Like it is also seen, but obviously there are some rare cases of it, right? So not rare cases, but very less cases. So I hope this is clear to you. If you have any kind of queries, you can ask me in the comment section, and I will be very glad to answer you guys.